All right, so if the DNA actually gets changed, this is DNA. All right, then, first of all, can you predict the parent code, okay, that actually coded for these chains of mRNA? Okay, we can predict that if we actually know mRNA, right? So, just 3 to 5. Okay, then whatever that coded for this mRNA is going to be AAA, not U, T, TTT, CCG, and then AAT, like this, okay? So that this is going to be actually code for this mRNA. But when actually this sequence changes, let's say, all right, so this one sequence actually changes to something random, G, all right, then whatever that gets transcribed here, instead of A is going to be C, all right? So before this got mutated, this was AAA, all right, but now it became CAA, which is a completely different triplet code, okay? So if it is AAA, remember there was lysine, AAA, yeah, so that was lysine, but then if you have CAA, then that's gonna be CAA, that's gonna be glutamine. Okay, so whole different amino acid will come along in a polypeptide chain, all right? So that is very dangerous because when you have different primary sequence, okay? If your primary sequence actually changes to different sequence like this, okay? Then your folding of DNA actually gets changed. Your tertiary structure actually changes because your identity of one amino acid changes, okay? So your entire protein function will be lost because your tertiary structure becomes whole different from whatever that has to be produced. You know what I mean? So that is very dangerous. Okay, so that's on page 140. So that's gene mutation. So this is called point mutation, all right? So there's one point where the base was actually substituted. So that is point mutation, or that is a base pair substitution. So when you go to an example, it says normal, the fat cat saw the dog, okay? But only one letter gets changed. So the fat cat saw the hog, okay? And it means the whole different meaning, okay? So, one example is on page 141, all right? Okay, before we look at this, let's look at a picture for this, for example. Okay, so again, if you can't read this, okay, please refer to the printed version of this. Print this PPT out, okay, and then look at this as we go along together, all right? So when you look at the first one, all right, so these are the triplet codes that code for these kind of polypeptides, okay? But if there's a base substitution in DNA, so instead of CTC, if there is CAC, right? So if this actually is changed to a different pair of base, okay? Then A actually gets transcribed into U in mRNA. You know what I mean? Then mRNA actually produces whole different polypeptide chain because this triplet code GUG actually codes for G, U, G. So that actually codes for valine, right? So instead of glutamate, actually valine gets formed, okay? So in that case, this polypeptide chain becomes different from this polypeptide chain because primary sequence is different. So then it's going to cause misfolding of protein, and that's going to produce whole different molecule of protein. You know what I mean? All right, so if another mutation occurs, so other than substituting, okay, deletion in DNA occurs. So this T actually gets eliminated, and then instead of CAC, CTC, it's going to be CCCTC, okay, without A, all right? Then what happens is your whole frame of reading ribosome is going to be changed, okay?